All right, welcome back. We are talking about chapter 20 today, which is all about phylogenies. And this chapter will discuss some of the basic background. What is a phylogeny? What is phylogenetics? We'll talk about what kind of information can be used to create phylogenies, how you could use a phylogeny, and then at the end of the chapter, we'll tie this whole idea of phylogeny into biological classification, which is something you may be more familiar with. All right, so what is phylogeny? So it is, in short, just the evolutionary history of the relationships between a particular group of organisms that you want to study, and it's typically portrayed in what's called a phylogenetic tree, which would be a diagram similar to what you see in the on the slide here. And a phylogenetic tree is basically a hypothesis in terms of how this specific group of organisms that you are studying are related to each other. Based off of the best available information, this is how we think these organisms are related, which ones are more closely or more distantly related to each other. And that can be very useful information to have, right? If you're a biologist studying these particular organisms, it may be very useful to know, oh, yes, these two are very closely related, and these two are more distantly related. And there are some very practical things that you can do with this information that we'll get into a little bit later in the chapter. Um, but if you're unfamiliar with phylogenetic trees, you're always going to have the group of organisms that you're studying on the right. So this is a very simple phylogenetic tree with just four organisms: chimps, humans, gorillas, and orangutans. You'll have the organisms on one side of the tree. At the other side, at what's called the root of the tree, you're going to have some common ancestor that gave rise through evolution to all of these current organisms. And it is very likely, because you're talking about a very long time scale typically here, that this common ancestor may be extinct at this point. Another key characteristic of these trees are these branches or splits, which are technically referred to as nodes. And each one of these nodes or splits is going to represent some significant divergence in lineages, some speciation event that gave rise to these new lineages that then, through additional speciation events potentially and evolutionary changes, ended up at the organisms that you're studying today. And you'll also notice, if we look at the bottom of the tree here, um, we're assuming in most cases the organisms that we're portraying in this tree are present in current day. And as we move back down the branches, we're going back in time, in many cases, millions of years, right? Because evolution typically proceeds very slowly. And another key thing to note is you can really rotate around these nodes. So the vertical order here is somewhat arbitrary. And what I mean by that is, for example, we could rotate here around this node at the split between chimps and humans. So we could draw it here, like you see on the left, where chimps are on top and humans on bottom. And that would be essentially identical to it being drawn as you see on the right, where humans are on top and chimps are on bottom. So, this image on the left, you would not interpret this to say, oh, okay, well, humans are more closely related to gorillas than are chimps because they're sitting closer together in this image. Um, we would just say chimps and humans are both basically equally distant from gorillas. So that time scale that you see there, how do we get that? How do we insert this element of time into a phylogenetic tree? And there's really two key ways you could do this. So one is through using fossil evidence, right? So as I mentioned, the common ancestor and maybe some other species that might be present, ancestral species that might be present throughout this tree that are not explicitly being shown here. may be represented in the fossil record, right? And we can date fossils very accurately in most cases. So if we have fossils representing some of these uh, sort of transitional species that may be present in this tree, this can give us a sense of time. And another more modern way to do this is using what are called molecular clocks. And this would only be possible if you have some genetic data on these different organisms. But basically, what a molecular clock is are specific gene or protein amino acid sequences that have been identified through a lot of research to change at a very slow, steady rate over time. So, if you can identify one of these particular genes or proteins, you can use that essentially as a molecular clock because it's been established. Yes, this particular sequence changes very slowly, changes very consistently, and you can basically observe those changes happening and use that to help set your time scale. So really, if we think of the big picture in terms of phylogeny, really the goal of phylogenetics would be to try to assemble the complete tree of life, the complete evolutionary history of all life on Earth. And you would end up with some, huge, incredibly branched tree that looks something like this, where this point down here is representing the origin of life, the very first living organism on Earth. And we go through many, many nodes and branches and splits, and eventually giving rise to this huge diversity of life on Earth, from the bacteria and archaea to single-celled eukaryotes and all the plants and animals and fungi that we know today. Humans would be up here right at the very tip of this tree. And so this would be the goal. It would include, all organisms that have ever lived on planet Earth. But of course, we're probably never going to get to the point of having that complete picture, um, just because, the vast majority of organisms that have ever lived on planet Earth are now extinct, and the fossil record is very fragmentary, and so we're probably never going to have a full picture. But that doesn't stop biologists from trying, and we certainly do have a lot of detailed information at this point, so we can draw a very large and complex tree of life. All right, so as I said, right, this phylogeny is basically a hypothesis in terms of how this group of organisms are related to each other. Um, and traditionally, the common way to do this was just by comparing physical characteristics and seeing which organisms share the most in common in terms of their phenotypes, their morphology, the idea being that the more similar two organisms look, probably, not always, but probably the more closely related they're going to be. Um, and so there's this important idea that comes up in the field of phylogeny, which is the idea of homology. So you could say these particular traits are homologous between these two organisms. Um, so, for example, um, you know, let's say primates and rodents both have ears, right? So that's a trait, a character that these two groups share with each other. And we would say that particular trait is considered homologous if the data suggests that both of these organisms have ears, because it's likely that they had some common ancestor back in time that also had ears. And, depending on what the data in your evolutionary tree, your phylogenetic tree shows, that may or may not be the case. But we would define homology as similarity due to common ancestry. Um, this particular phylogenetic tree that we see here is what's called a cladogram, and if we're looking at just the traits, the physical traits that we're considering in this tree, we can see these marked here. So these are the characteristics that were being used to assemble this tree, right? Do these organisms have vertebrae? Do they have a bony skeleton? Do they have four limbs, etc.? Um, any trait that you see in a tree like this that is basically present in all of the organisms, in this case something like vertebrae, right? These are all vertebrates. We would consider this to be the ancestral trait for that particular group. And as that term implies, uh, we would argue that that trait has been around for a very long time, right? Because it's present in such a large number of organisms. Whereas any of these other traits that are not present in all of the organisms, but only some, so for example, four limbs, right? Basically, all the organisms above this point, so amphibians, primates, etc., have four limbs, whereas those below, the fish and the sharks, do not. Um, any of these traits that come into play above the ancestral trait, we would consider derived traits. And that's just simply meaning that they have appeared more recently in evolution. And, that's a relative term, right? Relative to the ancestral trait. It may still have been millions of years ago. But essentially, what you're seeing is as we progress through evolution, we're seeing more and more newer derived traits appearing in organisms, typically getting more and more complex through evolution. So, in contrast to homology, traits could also be considered analogy. And the idea with analogous traits would be a situation where two organisms share the same trait, but the phylogenetic tree does not suggest that they shared a common ancestor that also had that trait. So, kind of a classic example of this are the wing flaps that are shared between sugar gliders and flying squirrels. You're probably familiar with flying squirrels. They live in Kansas. You find them around here in southeast Kansas for sure. Whereas sugar gliders, if you know these, you may have seen them in pet stores. They're sold pretty commonly as pets. They, have a very similar body form to a flying squirrel, but they're actually marsupials, right? More closely related to, kangaroos and koala bears. Um, so, yeah, sugar gliders and flying squirrels do share some common ancestor if you go far enough back in a phylogenetic tree, but it's almost assured that that common ancestor did not have those unique wing flaps, right? So, because the common ancestor doesn't have that particular trait, we would say the wing flaps are analogous and basically evolved independently in these two different groups, in these two different species. So, bottom line, homologous traits, there would be a common ancestor that also had that trait, whereas analogous traits, there would not. So, in terms of how these trees get constructed, um, the best, quote unquote, best phylogenetic trees are going to be those that use the most data. So, you would want to look at hundreds or even thousands of different physical traits and compare those between your group of organisms. You would want to maybe even include like DNA sequences. So, really, the more data you could pour in, the better. Um, and typically, you're going to feed all this data now these days into a computer program that's then going to assemble the tree for you. But these programs are always going to use what's called the parsimony principle, which is essentially just the idea that the simplest explanation is usually going to be the best. And in terms of evolution, the simplest explanation is going to be the one that has the fewest number of branches, the fewest nodes, because those are going to be very unique events. These splits that result in different lineages are typically going to be quite rare. Um, so if you can come up with a logical way that these organisms are related, that fits all this data and these thousands of traits that you're comparing, that has a fewer number of branches than another, that version with the fewer branches is probably going to be most accurate, path of least resistance, so to speak. Um, so what kind of data are you going to use? Well, as I've already mentioned, the more data, the better. And traditionally, biologists were using morphological data, right? Morphology, just physical traits, right? Do they have ears or not? Do they have tails or not? Do they have vertebrae or not? All those kinds of morphological data are very useful. In some cases, you can use fossil data. I already mentioned that as well, right? And that's particularly useful in terms of trying to uh, establish a time scale on our phylogenetic trees. Developmental data could be used in many cases, molecular data, DNA, protein sequences, and rarely, but sometimes, behavioral data. And I will link to this video that gives a very nice illustration of how some of these different types of data can be used to piece together the evolutionary history, phylogeny of one particular group, in this case the cetaceans, whales. But I encourage you to check that out. So what I want to do is just run through some of these different types of data real quick. Um, and we've already talked a bit about morphology, so I won't belabor the point here. Um, but um, again, this is the sort of the classical way that phylogenetic trees were constructed. So just picking your group of organisms, picking a group of traits, and basically just comparing to see uh, who has those traits and who doesn't. And typically, you would create a big table where you're kind of using sort of a binary code where you would put a 0 if a particular organism does not have a certain trait and a 1 if they do. And then you would basically organize the organisms from those that have the fewest traits, branching off the farthest back in time, to those that have the most traits, being sort of at the top of the tree. And so there are some limitations to using morphology, right, physical form, to construct phylogenetic trees. So, there are some groups, or what are called taxa, that have very few morphological differences, makes it difficult to compare. And some morphological differences can be caused by the environment. So that can confuse the issue, because really, when you're talking about evolutionary history, you only want to focus on things that are genetically determined. So anything influenced by the environment is not going to be very helpful. Development was another key type of data that can be used to construct phylogenetic trees. And what this is referring to is actually looking at the developmental stages of the particular organisms that you're including in your tree. And so, a good example of this, let's say you're creating a tree that contains both sea squirts, which are marine invertebrates, and then tree frogs, so obviously a vertebrate. If we look at the adult organisms and we're just comparing their morphology, we're going to find almost nothing in common there. So that's not going to be very helpful. But if we look at the developmental stages and compare those between a sea squirt larva and a frog larva, all of a sudden now we do see some structures in common. So they both have what's called a neural tube and a notochord. So there is some similarity there, which might suggest some common ancestry at some point, right? So by including that developmental comparison, we're getting some useful information that we would have missed if we were just looking at the adults. Fossil evidence, as we discussed, can be very helpful in terms of establishing that time scale, and is also giving us some information on maybe some of these transitional species that may or may have existed and given rise to some of the current organisms that we're studying in that phylogeny. And really, the biggest limitation of the fossil record, as I said before, is that it's pretty fragmentary. So, fossils form very rarely, and of course, they're buried in the earth, so they're difficult to find. Um, so, and easily destroyed in some cases. So we're kind of limited in terms of the number of fossils that are available. But we are finding new fossils all the time. So eventually we're getting a more and more complete picture. Behavior, as I mentioned, can be used occasionally. Um, the thing to keep in mind, right, is we only want to use genetically determined behaviors in constructing phylogenetic trees. And so a lot of behaviors tend to be learned. So, for example, bird calls or songs are typically learned and are not going to typically be very useful. Whereas, interestingly, frog calls are often genetically determined, and you might be able to use the specific sounds that a frog is making as part of the data to creating your phylogenetic tree. And really now the most modern and one of the more commonly used types of data these days are what we'd call molecular data, being primarily DNA sequences or the amino acid sequences of proteins. And that makes a lot of sense, right? If your DNA is the instruction manual for building the organism, it makes sense to argue that, okay, if two organisms have more similar DNA sequences, they are probably more closely related than two organisms that have very different DNA sequences. Um, and as we sequence more and more genomes, right, we have just a huge amount of data that we can use um, in helping us to construct these trees. All right, so hopefully you have some basic understanding now of what a phylogenetic tree is and some of the different types of data that could be used to create it. But really, this comes back now to the question of, well, what is the use, right? And on a basic biological sense, it's pretty obvious if you want to study these organisms or study life on Earth, right? You want to know something about how those living things are related to each other. Um, but there are some more practical things you can do with phylogenetic trees. So you can make some predictions about extinct organisms. Of course, you're learning about the evolutionary history of, let's say, complex features. Like the function of let's say, the human eye, for example, right? We can observe the evolution of the eye and learn something about how that structure actually works. Um, we're learning more about the order and diversity of evolution as well. Um, but I wanted to focus in on one sort of biomedical example in terms of making predictions about really pretty poorly studied species. So this particular example deals with the commonly used chemotherapy drug called Taxol, which was originally produced from this tree called the Pacific yew tree. Taxol is a drug that disrupts the mitotic spindle. It's a pretty, as I said, widely used and very helpful chemotherapy drug. But because there's not a lot of Pacific yew trees around, right, it was pretty difficult to get and making it very expensive. Um, and originally, researchers were having trouble synthesizing Taxol in the lab for many of the compounds they were getting out of Pacific yew trees. So, what researchers did was turn to phylogeny and looked at a phylogenetic tree to see, well, what are the other closely related species? And maybe we can identify something closely related to the Pacific yew that might allow us to um, find either some similar compounds or some precursor compounds that we might be able to use to synthesize Taxol more cheaply. And sure enough, by looking at the phylogeny, that pointed researchers towards the European yew, um, which contains some compounds that were able to be used very efficiently to synthesize Taxol in a laboratory setting. So now it's cheap, quote unquote cheap, right, for chemotherapy treatment, and is a very widely used chemotherapy drug. So the last thing I wanted to focus in on is this whole idea of phylogeny tying in with biological classification. And so if you've ever taken a biology class, you may be familiar with the binomial nomenclature, right? Every individual species has this two-part Latin name, right, that's intended to be very unique and unambiguous. The first part of the name is the genus, the second part is the specific species epithet. But this really all comes from the idea of phylogeny and relatedness, as we'll see. So there's this general term taxon, and or the plural of that is taxa, and taxa are just any group of organisms that is treated as a unit for some logical reason. So let's say we've got our phylogenetic tree here, includes a diverse group array of organisms here. We could group together everything in this particular tree and say, okay, we've got a taxon here, and we're gonna consider this group the vertebrates, right? Because they all have this particular trait they share in common. Or within that larger group of vertebrates, we have this subset here that includes mice and chimpanzees that we're going to call the mammals, right? It's another group that we can lump together because they share some things in common that others do not. So the whole idea is you can take your phylogenetic tree and start basically grouping and organizing organisms together for logical reasons. And those groups really are the basis of classification. And there's certain things that make a quote unquote good taxon. So usually uh, evolutionary biologists want taxons to be what are said to be monophyletic, meaning the group contains all the organisms that should be grouped together and the common ancestor of those organisms. So this would be an example down here in the blue box of a good monophyletic taxon. So we've got these three species, H, I, and J, that are grouped together for whatever reason, and this group extends all the way back to the common ancestor, which would be here, of those species. And you can usually identify a good monophyletic taxon because you can remove that whole group by just cutting a single branch on the tree. And there's a couple of common mistakes that happen. You can end up with what are called polyphyletic or paraphyletic taxons. So a polyphyletic taxon is one that does not include the common ancestor. So we see this group here, right? We really needed to extend this group all the way back to include this common ancestor back here. Whereas a paraphyletic taxon, it includes the common ancestor, but it's missing one of the descendant species. And you might look at these and say, well, duh, why didn't they just draw the box like they should have? Well, usually you're ending up in one of these situations just because there was some information that was missing, right? So, for example, maybe we just discovered species A, or we just discovered it had some trait that we didn't realize it had, and so it's just been positioned here now in this tree, whereas previously it was not included in this group. Now we realize, oh, it should be. So it's usually just a mistake due to lack of information. And this really points to the fact that you have to constantly reconsider and redraw these phylogenetic trees, right, based on the most recent evidence. So they are just hypotheses, and those hypotheses are constantly changing. And so if you've ever studied classification before, you may have seen the hierarchical classification, right, which is the whole idea that you have these sort of nested groups from the three domains of life, bacteria, eukarya, and archaea, that are the three really broad groups, down to specific individual species that each belong to an individual genus, that each belong to an individual family, and so on. So I do want you to know the order of these nested groups from most broad to most specific, and there's a number of different tricks that you can use to remember these. But take a few minutes and see if you can drill this into your head from domain to kingdom to phylum, class, order, family, genus, species. And while we're talking about classification, there are some very specific rules that are used in the scientific community to assign these particular names. And the whole idea here is we want to make sure that there's only one scientific name for any given taxon, right? We don't want any ambiguity there. And historically, there was some confusion because you basically had the zoologists kind of with their own set of rules, and in terms of naming, and the botanists were doing their own thing, and the microbiologists their own thing. So in more recent years, these groups have sort of gotten together, and we've tried to apply a more uniform nomenclature. So there's been some renaming and things like that have taken place to try to make things a little bit more uniform. All right, that is all I've got for you.